Hi, my name is Magdalene Spitzer, and I will be giving a presentation today on the gastric phase of digestion. So there are three phases of digestion. The first is cephalic, but we'll be focusing on the gastric phase, which is then followed by the intestinal phase. During each of these phases, there's a secretion of gastric juices that can be stimulated or inhibited. So this presentation is just going to focus on the gastric phase, and this is where 2/3 of these secretions occur. Okay, I just want to overview the stomach first. The stomach is the site of the gastric phase of digestion. The stomach has three main functions, and those are storage, digestion, and defense. For storage, the stomach stores food and regulates its passage into the small intestine. If you remember from our lectures, there's the pyloric sphincter that controls how much of the stomach contents can enter the small intestine at one time. So it's very regulated, and the small intestine is actually where most of the digestion and absorption occur. The second role of the stomach is digestion. There's some digestion done in the stomach. The stomach chemically and mechanically digests food into a soupy mixture of small particles that we call chyme. The third function is defense. This has two meanings. So the stomach first protects our body by destroying many of the bacteria and other pathogens that are swallowed with the food or trapped in the mucus that makes it through our airway. And then the second role of defense is actually the stomach protecting itself from its own secretions that can be very acidic and damaging to the epithelium. Okay, how is the gastric phase initiated? The vagal reflex during the cephalic phase initiates digestive activity before food even enters our mouth. Once food enters the stomach, there are stimuli in the gastric lumen that initiate a series of short reflexes during the gastric phase. So one of the biggest components is the distension of the stomach. So stretching of the duodenum, which is the first segment of the small intestine, enhances the gastric function through the vagal nerve again. So the chyme causes secretion of gastrin, which stimulates the stomach. And then the acid and the semi-digested fats in the duodenum trigger the enterogastric reflex. So the duodenum sends inhibitory signals back to the stomach through the enteric nervous system. And then third, the newly arrived chyme also stimulates the enteroendocrine cells of the intestine to release compounds that can stimulate some accessory organs like the pancreas and the gallbladder. But they can also suppress gastric secretion and motility, which allows the duodenum to process the chyme before it receives more stomach contents. And this gastric phase can actually continue for 3 or 4 hours while the acid and the enzymes are being processed and helping process the ingested materials. So just to quickly summarize again, the stomach stretches and the pH of the stomach contents increases, and this causes a cascading series of events that leads to the release of HCl by the parietal cells, which we will get into in the next few slides. So gastric phase reflexes. Gastrin stimulates contractions in the muscularis externa of the stomach and the intestinal tract. There are stimulations of stretch receptors and chemoreceptors, which is what we were mentioning when we talked about the stomach stretching and having that distension. And then short reflexes are coordinated in the submucosal and myenteric plexuses as well, which activates the stomach's secretory cells, which we will go into a little more detail on the next few slides. Additionally, the myenteric plexus also produces powerful contractions that cause mixing in the muscularis externa. And in this picture on the slide that you can see, there's a good illustration of the pyloric sphincter, which we mentioned controls how much stomach contents enters the small intestine. And then you can see the rugae of the stomach, and then the esophageal sphincter at the very beginning of the stomach. Okay, this is just a little illustration here to show you all the different gastric secretions that occur in the stomach. So there's about 9 of them on the right side, but we can briefly summarize which secretory cells secrete them. So mucus cells secrete bicarbonate and mucus. Parietal cells secrete gastric acid, also known as HCl and the intrinsic factor. Enterochromaffin-like cells secrete histamine, and chief cells secrete pepsin, the precursor to pepsinogen, and gastric lipase. Gastric lipase plays a key role in the digestion of fats. D cells secrete somatostatin, and G cells secrete gastrin. So I will go over a little bit more what each of those secretions does, but those are the cells that are responsible for releasing them, and each of them are triggered by a different event. While we're on the side, we will just go over a little bit of gastrin and the role that gastrin plays. So there are several hormones, neurotransmitters, and paracrine molecules that influence the motility and secretion that occurs during the gastric phase. But when we look at gastrin, we can see that neural stimulation and the presence of peptides and amino acids in the chyme of the stomach stimulates the gastric secretion. So that's the trigger for gastric secretion. Gastrin then travels through the bloodstream to the parietal and the chief cells, and those cells then increase secretions to lower the pH of the gastric juice, aka making it more acidic. Also, gastrin does stimulate gastric motility. And as this slide showed you, gastrin is secreted by G cells, and they're regulated by short reflexes through the enteric nervous system, as well as the gastric releasing peptide, also known as GRP. Gastrin promotes the release of HCl from parietal cells, as we mentioned earlier. If the pH in the stomach is below 2, then the stomach inhibits the parietal cells and the G cells. And this constitutes part of the negative feedback loop that winds down the gastric phase as the need for pepsin and HCl declines. So that was a lot, but that's a little bit about the role of gastrin in digestion. Now here is a little summary of the role of the other secretions that we saw on the last slide. No need to memorize this. Like we mentioned, gastric lipase plays a role in digesting fats, and pepsin, the precursor, and then pepsinogen, they have a role in digesting proteins as well. And then 2 other things I would like you to notice on this slide. First, the opposing relationship or the roles of somatostatin and gastrin. So somatostatin inhibits gastric acid secretion, while gastrin stimulates gastric acid secretion. So they act in opposing ways, inhibitory and stimulatory. And then the other thing to notice is the role of mucus and bicarbonate. Both are protecting the epithelium from getting damaged, and that goes into a little bit of the role of defense in the stomach, which we will explore on the next slide. The gastric phase of digestion requires a balance of defense and digestion. So the stomach must protect itself from autodigestion by acids and enzymes. And they do this through the mucus bicarbonate barrier, which is created by the mucosal cells, which we saw in that last slide. Those mucosal cells secrete mucus and bicarbonate, and they form a protective layer to protect the epithelium from damage from the acidity of the contents of the stomach. This protective layer can fail, though, which causes the formation of ulcers. So normally, most commonly, ulcers are caused by NSAIDs, like your pain relievers, Advil, ibuprofen is an example, or certain types of bacterium that can cause inflammation. However, there is a specific syndrome called Zollinger-Ellison syndrome, which is due to the secretion of excess levels of gastrin. So that's more rare, but that can also cause the formation of ulcers. And then once the chyme passes into the small intestine, the gastric phase of digestion ends. We mentioned previously there's that role of the negative feedback loop. So with the end of the gastric phase of digestion, the next phase would be the intestinal phase of digestion.